Good afternoon, students. I hope I'm audible.
Good morning, ma'am.
Good morning. So that means I'm audible, right? So we are going to start this module about plant disease management now, right? So in the last module, we studied about the pathogenesis part, and we studied that how different host factors and then how different pathogen factors are there, which see pathogen factors will try to lead to the development of the disease, right? And the host factor will try to prevent the disease uh, to happen in the host. So we studied all those factors, the toxins, the how toxins and how different enzymes are being released by the pathogens, and then the host factors are maybe the resistant factors in the host, or certain chemicals or phytolexins or compounds which are being released by the host, or the hypersensitive response or SAR resistance. Right? So there are factors which are involved both ways, right? So management, I'm sure you are, and you are aware of this term, because when we studied the virology course, we discussed the management, a lot of management there, right? So diagnosis and management was the important aspect there also, and the similar thing you're going to study in this, but in a more detail, because in virology we still have some of inhibitions, and there are certain things that uh, very less. We don't have antibiotics, for example, for the viral diseases. We cannot use. Then in since this is plant virology specifically, we are talking in plant virology also. Diseases cannot, or plant viruses cannot be managed once they are already in the field, right? So we cannot do much after they are established in the field, but we can do a lot of things before they are there in the field, right? So preventive measures, right? So prevention is better than cure in case of a plant viruses also. That lot of preventive steps we can take so that the pathogen is not being introduced into the field, right? Vector management can be there. We can, crop rotation can be there, or we can just change the time of sowing a particular crop in the field. So many things we can do, right? Similarly, in case of the fungal pathogens or a bacterial pathogen, we still have advantage because we have a chemical control also, right? Because fungicides are available. Then for insects, we have the insecticides, right? Certain in plant, we don't use antibiotics in the plant in the bacterial diseases, but yes, some such as tetracycline can be used in a bacterial diseases in the plant, right? So we have a lot of management practices available in the plant pathogen, especially for fungi and bacteria. So the first important thing is that why management is required. Management is required because we don't want the losses in the crops which are being caused by the pathogens, right? So this is that is why a management is required. And then the second, another thing in the management is that we don't depend only on one particular method of a management, right? We discussed the integrated disease management last time also. So similarly, in other plant pathogens also, the integrated disease management is being implemented, right? So that means we are not using only one method, but we are using every available method. It could be the biological control, it could be the culture method, it could be some resistant plants against the pathogens, right? Or maybe burning of the residual crop plants in the field, or maybe the crop rotation, fallow of the land, just leaving it like that. Soil solarization could be there, right? So a lot of methods combined. And why integrated disease management? Another question is that why we want to do that? Because we want to discourage the use of chemicals in the field, right? Because from the past many years, a lot of studies are being done, and the amplified use of chemicals in the field, maybe in the form of fertilizers, in the form of insecticides or fungicides, has led to the development of a residual problems, that these pesticides, they are somewhere, stay there in the plant or stay there in the food, and when we consume them, even after even we wash them, it somewhere goes inside our body, and a lot of cancers and a lot of other diseases are being reported to be caused by this. this kind of a problem. So chemical control is discouraged and only used in those cases where it is actually required, and also in a proper concentration, education of the farmers so that they don't misuse it or overuse a particular chemical in the field, right? So that is important. So this is why the integrated approaches are being followed so that we can reduce. And another aspect is, like, you all must have heard about the organic farming these days, right? When you go to the market, so there's a separate corner where they'll say that these are the organic products. So what does the organic products means? Organic products means that no synthetic chemicals or maybe any fungicides or insecticides are being used in that particular field, right? And this concept has also come up because a lot of diseases are being caused due to the extensive use of these chemicals in the field. So due to that, people are moving back to some culture where we least use them or where we don't use them, right? So this is somewhere very important, right? So that is why we are moving towards, or we are going where the least chemicals are being used in the field, right? The problem arises where it is not the chemical use also, it is actually about the amount of the chemicals which are being required to be used in the field. Because what does farmers, usually farmers are a group of people where they are not much educated about these things, or maybe farmers, they may hand over these things to the employees, so they don't know how much amount to be used. So the main problem lies in the appropriate concentrations which are required to be used. And usually what they think that the more they will use in the field, the more their crop will be free from the pest or the pathogens, and their yield will be more, without thinking that this is causing a loss somewhere or health issues are being caused due to this. So these are also problems that we need to look. And there are several extension officers which do these kind of a work, at which there's a bridge between the universities or scientists and the farmers, that we need to educate them how to use these things, right? So today's lecture, I'll be just discussing about the basic principles. You have studied, I'm sure, मैंने last time भी ये आपको lecture करवाया था, but then we'll be studying these things in detail. For example, we'll study biocontrol in detail. We'll study other, like quarantine, what is the role of a quarantine. Then we'll also study the other cultural practices that we do. So all those things we are going to study in detail. Today we are just taking an overview. And the management part also comprises the structural defense mechanisms or defense mechanisms which are being used by the plant. So how we can use them also in the management practices, right? So okay, so plant disease management, as I told you, we uses an integrated approach where exclusion, these are basically called the principles of a plant disease management. Also, if there's a question like what are the principles of a plant disease management, so you're going to write this: exclusion, plant resistant, cultural control, and the chemicals. So exclusion means that how pathogen is removed from the field, is excluded from the field, right? Exclusion means excluded, कैसे हमने उसको field से निकाला, how we are avoiding the pathogen in the field. Then resistance means how we are using plant resistance in the natural resistance. Some plants are naturally resistant to some pathogens, so we can use those varieties. Or some varieties are being generated which are resistant to the pathogen, so how we can use those varieties. Cultural controls, there are a number of cultural controls such as soil solarization, it could be the crop rotation, it could be the fallow, to keep the land fallow for certain time, right? So a lot of cultural methods are also available. Certain cultural methods are also available to eradicate the insect pests, especially those are the vectors of the viral diseases. So all those factors, right? Then chemical control is at the end. So what are the different chemicals such as fungicides or insecticides or nematicides which are available in the market, which we use for the management of the or the control of a particular disease in the field, right? So exclusion means regulation of plant materials at the ports, city, country, state or country boundaries. There are certain federal and state rules. What does this mean? This means that there are certain there are certain quarantine agencies at the national and international level which are very active in these things, right? So what does this mean? This means that it's on our own. We cannot just transfer any kind of a material, whether it's a planting material or a seed, from one place to the other place, or one country to the other country. So they are being checked by the quarantine agencies that they are free from the pathogens, or we also need to provide a certification that we are free from any kind of a pathogens. So those are called quarantine agencies or a quarantine rules. So yeah, in India, there's a National Bureau of Plant Genetic Resources. Its headquarters is in Delhi, NBPGR. So it deals with these kind of a thing. So there are certain guidelines which are being laid down by these agencies that on our own we cannot move a planting material, because with see what happens, there are 90 to 100% chances that if a plant is affected or infected by a pathogen, that pathogen is going to move to the other part also where it is not available already, right? So it might find a conducive environment also there, or a good host also there, or some vectors also there. So it is a possibility that in the place where it was earlier, it was not causing much losses, but when it reached a new place, there everything is perfect for the pathogen to grow and it is causing losses. So this is why that we need to be very careful, right? So pathogen-free seeds or a planting material such as seed certification, right? So certified, it is on and on every time being, farmers are being told and they are being encouraged to take certified seeds from the those shops where there is certification there that okay, that seed is free from these pathogen, right? It has been tested. So how it is being done? It is being done in the referral labs or referral centers which are available throughout the India, throughout the country, that they'll be testing the seed lots for the presence or absence of the pathogen, right? Then meristem culture, we know plants which are being generated through the meristem tip culture or a meristem culture are free from the pathogen. And then cuttings from clean mother plants under dry. This is for the vegetatively propagated plants. So some plants we are grown from the cuttings, right? Stalks लेते हैं, उससे cuttings लेते हैं, and we can grow directly. Tubers also similar case. So those mother plants should be free from any kind of the pathogens, because if they are free from the pathogens, then definitely plants that we are growing in the field will be free from the pathogen at the initial stage. If they are already infected by certain pathogens, so we are carrying forward the pathogen directly to the field, right? So we need to be very careful in the beginning that how we need to avoid it. Then resistance, okay, the resistance could be the genetic, that some plants are naturally resistant to some kind of a pathogen and some plants are susceptible to the some pathogen. So we should be careful enough to select the resistant varieties, not to grow the susceptible varieties in there, or through the lot of breeding programs are being there, or a lot of plant varieties have been generated which are free from the pathogens through the conventional breeding methods also, and through the transgenic methods also these days. So this is an ongoing process, right? But
Ma'am,
then in order to develop, yeah,
ma'am,
hello,
how will yeah? Sorry to interrupt, ma'am. Ma'am,
no, it's
how
fine.
will how can we identify that a crop is susceptible to certain pathogen or not?
Okay, fine. See, for these things, when artificially, if we want to see, we are going to inoculate those crops or those varieties with the pathogens, and we are going to see whether what kind of a symptoms are being generated. So this is how we identify them, right? So basically, we do them. We do certain experiments in the field. We grow those varieties, we inoculate those varieties, and we see whether there are being some symptoms which are being developed, or which varieties were to least to develop the symptom, or where the symptoms were not developed, or which were the tolerant ones. So this is how we check them, right?
Yes, ma'am.
Okay. So this is again a very exhaustive process, but it is being done. It is being done since a long time, and we are still doing it. Yes, see what is the main point here, that it is not that if we are growing plant in the field, or a pathogen-free plant, so in the field it is not going to be impacted by any pathogen. So there are 100% chances कि अगर हम उस pathogen-free plant को भी अगर plant को grow करेंगे field में, so there are chances that it might get affected by the pathogen maybe at the later stages. But the benefit, what is the benefit there? because we are not introducing the pathogen, and at the pathogens also affect the plants at a particular stages. Usually the younger plants are mostly affected by the pathogens, right? So we get a time, right? Because the pathogen is not in the seed lot, it is free from the plant is free from the pathogen. So plant is growing there, right? Plant में field में आते आते उसको time लग जाएगा. So by that time it is usually there that particular stage is being crossed, or the losses which will be being caused by that particular pathogen, they'll be reduced, right? So basically हम totally protect नहीं कर पा रहे हैं pathogen free को start. Yes, resistant varieties अगर हम grow कर रहे हैं, then definitely those varieties in the later stages also in the field, they will not be get impacted by the pathogen. But there's a point here also. We cannot, one plant is affected by maybe 50 or more than 100, more than those pathogens, right? तो resist एक plant को हम हर pathogen के resistance नहीं बना सकते, right? This is a catch here, that we cannot do this. So, but yes, we need to see. Definitely all of those 50 pathogens are not causing 90%, 40% or 30% loss. उसमें से कुछ ऐसे होंगे जो symptoms आएंगे, losses नहीं होंगे, but only few will cause losses, right? So we need to select कि किस pathogen से loss ज़्यादा हो रहा है. So we should try to develop a resistant variety for this particular pathogen, right? So this is how this process works. So ऐसा नहीं है कि फील्ड में हमने एक पैथोजन के अगेंस्ट रेजिस्टेंट वैरायटी डेवलप के लिए से ग्रो कर लिया तो कोई और पैथोजन उससे अफेक्ट नहीं करेगा. नो, देयर आर चांसेस दैट अनदर पैथोजन इज गोइंग टू कम एंड अफेक्ट दैट पर्टिकुलर plant. But yes, since we have selected it wisely, we know that which pathogen is causing more losses. So we are working for those pathogens. Other pathogens we can still ignore, right? Then genetic immunity, we know. We studied this the last lecture also, that certain plants, they are resistant. They are naturally resistant to certain pathogens, right? So we can choose those plants also, or we can develop certain plants by breeding methods. Adaptation means plants adaptation to a site. So this is like basically when we are developing a new variety or in a plant breeding experiments. So plants which are being brought from the other place to a new place, so they are given some time to the adaptation to a particular environment. We test that, right? So after some time, those plants, they get adapted, well adapted to that particular environment, and there are chances that they are not affected much by those diseases which might be present in the earlier stages or earlier phases where they are. So plants adaptation to a particular environment or a site is also important. Then plant resistance, genetic. Okay, this is again we discussed this last time. The constitutive means the continuous differences which are the cell walls or a waxy epidermal cuticles or a bark leaflets, right? This we discussed last time. And indicible differences are chemicals or the proteins which are produced in response to invading pathogens, right? It could be the toxic chemicals or a pathogen-degrading enzymes or deliberate plant cell suicide, which means hypersensitive response, which is being shown by the plant or a host against the pathogens, right? Then systemic acquired resistance, also we discussed last time that this is again type of a resistance when a particular pathogen, it infects a plant, this systemic acquired resistance become activated, right? It is again long-lasting systemic immunity. Even it is found even in those tissues which are not infected. And it's a broad spectrum, and it is generally associated with the increase in phytohormone salicylic acid. So it's called in a particular plant, this assay, it increases and it gives a systemic acquired resistance to the plant. Then it could be the chemically induced also. Certain chemicals can also induce this systemic acquired resistance. What does that mean? So we can use some assay analog which can induce SAR-like responses in the plants, right? So they provide resistance in plant tissues, systemic resistance beyond the application site, and they are often referred to as a plant activators. For example, one is the ActiGuard, which is benzothiodiazoles, which is used for a plant protection. So such chemicals are also available which can induce such kind of SAR-like responses in the plant. It can be done in the field, right? Then biological induced, okay, systemic acquired resistance by a weak viruses. This is somewhere like a cross protection like phenomena. I guess last time I brought to the cross protection and plant viruses, right? Where we can just infect the plants with the weak or some non-virulent pathogens जिनसे ज़्यादा infectious नहीं होते हैं जो strains, and then these will provide immunity against the more virulent pathogens later stages, right? So again, then induced systemic resistance can be triggered by non-pathogenic plant growth promoted bacteria also, and it involves jasmonic acid ethylene rather than salicylic acid. तो इस केस में jasmonic acid से ज़्यादा ये trigger होता है, right? Then adaptation means right plant for the right site. For example, okay, this is something like in India we have a different climatic zones, right? We have a temperate areas, we have a tropical areas. So temperate areas are the areas where the temperature is low, the cold, hilly areas, whereas tropical areas are there areas where we are much hotter and the humidity is more in those areas, right? Then again, these types can again affect the pathogen behavior also, right? So there's one particular example which I discussed, I guess, earlier also, that the Begmo viruses, they were supposed to be the pathogens or viruses which affect the plants in the tropical areas, not temperate areas. But then later, they because it is transmitted by the vector which is called Bemisia tepici, or which is called a whitefly also. So this vector moved to the temperate areas to the new host also. So when the vector moved to the new host, the pathogen also moves along, right? But otherwise, yes, there is certain limitation that few pathogens, they are more found in the temperate areas or plants which are grown in that areas, whereas few pathogens they are found only in the plants which have been grown in tropical areas, right? Then cultural controls. So cultural controls are the cheapest one. They don't require much money, and they are the easiest one which farmers can do in their field, right? इसमें बहुत ज़्यादा educated people नहीं चाहिए. We just need to tell them this is how it is to be done and they can do it. So cultural control first could be the crop rotation. So crop rotation, as the name suggests, that we don't grow same crop in the same field year after year. Why? Because अगर आपका fungal या किसी उससे infected है plant, so there are chances in the form of a bacterial, in the form of a spores, fungal spores, or if it is affected by the fungal in the form of a fungal spores, it will survive. Maybe in the soil, if it is a soil-borne pathogen, or maybe the nearby plants, on the weed plants also, or other hosts also, right? So let's not grow the same crop in the same field, especially if it is affected by the similar kind of pathogen, right? So this is one factor. Then the alternate host eradication. This means alternate host means the other host plant. So when the original host or on the main host, primary host on which the pathogen attack is not available, it is going to stay on the alternate host. So if we remove this alternate host from the area, so they are not going to survive there, right? This can be done. Then sanitation of tools, equipment, potting. This is basically for the viral pathogens also, because they get transmitted by this way, though fungal and other bacterial pathogens also, but mostly the viral pathogens. So we should be very careful while we do these practices in the field. Then improved plant environment, water management, air circulation. So this means that the density of the plant in the field should be well looked into. The more the denser the plants in the field are, the more are the chances that they are going to be affected by the pathogen very easily, because the transmission becomes easy if one plant is affected. So we should know that they are not very dense. So a thinning of the plant is being done after the crop they become a certain height. So farmers, they do that basically. Nutrient management. So healthy plants are least affected by the pathogen, right? So there are chances, if their plants are healthy, so there are less chances that they will be affected by the pathogen. If they are already weak, there are lack of nutrients in the field, plants are already weak, not very healthy, so vigor, that means the overall vigor of the plant, it is not good. So they will be the first to get affected by the pathogen. Soil treatment such as soil solarization or a tillage. So soil solarization is basically a phenomena where the temperature of the soil is increased by the soil solarization. They just cover the field with a plastic sheet, right? Just before growing. They will cover empty field with a plastic sheet, properly covered. So it is going to trap the sun rays, or heat is being trapped. So temperature in that field will increase, right? It will increase. So that increased temperature will kill any kind of a nematodes or any kind of a bacteria or fungal spores if they are present in the field, right? So again, this is a phenomena which is being used, or mulches or other barriers. Sometimes we use barrier crops in the field. Maybe small crops, with them will grow tall crops in between, so that if they are transmitted by the vectors, the pathogens, the vectors, they are not able to fly very high, right? Insects, small winged insects. So they are not going to pass those barriers. So those crops are also grown in between as a barrier plant, right? Then chemical control. So chemical control में वायरिसाइड्स के अलावा आपको सब मिल जाएगा. You'll find fungicides, you'll find bactericides, you'll find nematicides, you'll find insecticides and fumigants. Fumigants basically आपके होते हैं जो fumes के through, मतलब they certain chemicals will generate fumes to kill the pathogen or vectors around the area. We use a fumigation for the mosquitoes also, right, during that season, rainy season, when mosquitoes live in the snow. So these are the fumigants. So we can go for a seed treatment, we can do soil treatments, we can do root drainages, we can disinfect the roots, right? So seed treatments are before the seed is being grown in the field. So that means if the seed they are carrying any sort of a pathogen or anything, so we can treat the seed with certain chemicals or just the hot water treatment. So that will kill the pathogen. Soil treatment, there are certain soil mulches or soil methyl bromide is basically most commonly, or other chemicals they come. So we just spray those, or we just drench the soil with those chemicals which are being used, so that it becomes free of the pathogen or any kind of a spore. Then roots also we can treat if we are just growing some plants, nursing a plant पहले grow करते हो, फिर उनको uproot करके then you take them to the field, right? In those plants, the roots can also be treated with certain kind of chemicals, or then disinfecting the tools. So this is basically trees में ज़्यादा होता है, because आप pruning करते हो trees, especially the perennial जो trees होती हैं आपकी, right? So you do pruning after every season before the next season comes. So they means that before, for example, you've done pruning in one tree, then you should disinfect that particular tool before you use that in the other tree also, so that the pathogens are not being moved. There are foliar sprays, so plants they are grown in the field, so certain sprays are available. Then trunk injections are there to provide help to some, to prevent some trunk diseases or some gall kind of a bacterial diseases, trunk sprays, or a post-harvest use. So this chemical control can be done in all these forms, right? So they say that for fungicides and bactericides, size is not accurate. They do not overall kill the pathogen, but they suppress the pathogen, right? पूरा उनको kill नहीं करते हैं, right? And as I said, there are no chemicals to use against plant viruses. So for plant viruses, we don't have anything, right? So in chemical control, there are a lot of fungicides. You just remember few, don't remember all. The copper sulfate is a very common one. जिनको cross लगाया है वो ban कर दी गई है. Arsenic, it causes arsenic poisoning in the soil, so it is being banned. Mercury chloride is also causes mercury poisoning, so it is being banned. Again, phenyl methyl, so cross जो मतलब है, they are not being in use, but other sulfur dust, copper sulfate, lime sulfur, borax mixture is very common for foliar pathogens, very commonly used, right? Then you leave this, you don't need to remember this, otherwise you will get confused. So इसको केमिकल कंट्रोल में ये आपको उन्होंने वो बताया डिफरेंट केमिकल्स का कॉपर कंपाउंड कितने परसेंट कहाँ पे अप्लाइड या यूज होते हैं, कितना परसेंट है, 23% सबसे ज्यादा आपका क्लोरोइंथ्रोल केमिकल्स हैं. कॉपर कंपाउंड उसके बाद कॉमनली यूज होते हैं, राइट? So these are the different compounds in a fungicides or a chemical control that we use. Then we have this one is interesting. We have a microbiological controls or a biocontrols also. So certain fungi and bacteria, they kill other fungi or other bacteria, right? So we can use them in a biocontrol or a microbiological control. For example, fungi, the trichoderma is the best one. Very cheap and very easily available. Then we have Pythium also, we have Verticillium also, and bacteria. We have Bacillus species, Streptomyces, Xanthomonas. All these can be used in a biocontrol. Pseudomonas is a very commonly used in a biocontrol. Agrobacterium. So all these can be used as a biocontrol methods also, right? Then this is just one example where the Bacillus subtilis strain, different strains of a Bacillus subtilis are being used as a fungus control to control the fungal pathogens, right? So this is again being used. Then, as I told you, that integrated approach which include exclusion, resistance, cultural, chemical, microbiological is being preferred as compared to the single approach or a single management practice. So we always lay emphasis that we need to follow the combined approach so that minimum use of like chemical should be there, right? So GMOs, with genetically modified plants, there has always been issues, there are societal issues or some ethical issues or some other issues that some group of people they do not accept GMOs, right? They have their own thoughts that we are genetically modified plants, especially if we are introducing genes from maybe other kingdom of, for example, from bacteria to the plant or from these things. So they say that we are violating the natural process somewhere and we are polluting the genome of the plants also somewhere, some many issues. You'll see it. Just Google it, right? What are the this ethical and societal issues with the GMOs? But yes, I would say here that the genetically modified plants, we are developing as a for a pathogen resistant are definitely or are actually being helpful in the management of the plant pathogens because as I told you, right, breeding experiment or a breeding for a virus or a pathogen resistance is a long process and even at the end of the sometimes process we come to know that plant is not that resistant or maybe the plant is resistant but now its yield is not as we wanted. So again it has its own pros and cons, right? So we cannot depend, but genetically modified plant or a plant disease management is definitely good option and has proved to be a good option, right? And then we have other methods such as the cross protection method we use for plant viruses, plant pathogen derived resistance, for example, both protein mediated resistant or a movement protein resistant is being very commonly used in the plants. It's been done, right? So number of methods are available, but then again, what is the point? Important point here that the economic, how economic they are because farmers they're coming from they cannot spend much. For example, the biological controls they are also not very cheap somewhere. Not all are very cheap. So definitely not all farmers they're going to afford this kind of a control, right? So we need to look for a method which are cheap, which are economic to the farmers so that they don't hesitate and they don't have to spend much money on buying these things in the field. Fungicides and insecticides they are very cheap prices because a lot of companies are there which are getting benefit out of this. So these things are being sold at a very less rate. So that is why they afford it and they buy it and they use it as much as they want to use in the field. So this is causing a lot of trouble and a lot of problems. And beside this, we have a certain botanicals also. Botanicals means certain plant extract which repel certain insects or which reduce certain diseases. So those are again not much widely used, but yes, a lot of research papers are available where people have tried different extracts from the plants which can repel the pathogen or which can reduce the growth of a pathogen in a particular. So that those are again methods that we need to work more on those methods. We need to work on more on environmental friendly methods because these fungicides and all those things they go to the soil, they reach the water table, they are polluting the water bodies where people are drinking water, where animals are drinking water. So they're causing a lot of trouble. Somewhere a lot of health hazards or health related issues. So we need to come up with the methods which are chemical free. So genetically modified plants are definitely answer to these things, but then again we need to see that how much they are being accepted or they are widely accepted or not. But in US and many other countries, the genetically modified plants are widely accepted. The corn, the canola plant, maximum they're all genetically modified plants they use there. So that kind of approach is to be required to be followed in all other countries also, I guess. So definitely it will help a lot in the management, right? So I think we'll stop here for today, and then one by one we'll try to take in detail the biocontrol maybe in one lecture, or the other cultural practices in detail in one lecture. So we'll try to take up one by one each of them, right? So till now, if there's any confusion or anything, just speak up. Fine. Seems like you've understood also. That's good. So just go through this also. I'm sure the management part will not find difficulty because it's kind of a repetition. So fine. That's all from my side. I'll see you tomorrow in the next class. Before that, I'll just give you attendance, please. Yes, Adish Mishra, please switch on your cameras and give your attendance. Adish Mishra,
Present, ma'am.
Athoiba,
Present, ma'am.
Ayushi,
Present, ma'am.
Sam, Devanshi,
Present, ma'am.
Ishita.
Present, ma'am.
इशिता के बाद है जानवी.
Present, ma'am.
क्षितिज. Okay. And Manaswani,
Present, ma'am.
Namrata,
Present, ma'am.
and Namrata के बाद है कौन? नंदिनी राज. Nandini,
Present, ma'am.
Nehal, Rupal. Nehal, I can see. Rupal, I can't.
Present, ma'am.
Sahiba,
Present, ma'am.
Sanjana,
Present, ma'am.
and we have Shakti,
Present, ma'am.
Shivani,
Present, ma'am.
Tanvi,
Present, ma'am.
Vidit, Vivek,
Present, ma'am.
Yukta,
Present, ma'am.
Yuvraj.
Present, ma'am.
Okay, guys. Okay, guys. Thursday, Thursday we have a presentation. We have two presentations this Thursday, right?
Yes, ma'am.
Okay, fine. I'll let you know before that only. Fine. Okay, I'll see you tomorrow then. Thank you. Bye bye.
Thank you, ma'am.